Okay, hi everyone, welcome to the session. This is the fifth lecture of MD6028. What we're going to talk about today is the genome expression and replication of group 6 and group 7 viruses. And when I'm saying group 6 and group 7, I'm referring to Baltimore classification. So in the last 2 weeks, we've discussed genome expression and then genome replication, but we did so for the first 5 Baltimore classification groups, right? We did so for the class 1 to 5 viruses, and we kind of ignored group 6 and group 7, and this is what we're going to address today. So we're going to talk about both genome expression and genome replication for group 6 and group 7. It makes a lot of sense to do things this way because group 6 and group 7 are slightly more complicated than the other groups in terms of how they express and replicate their genomes. So it just makes a lot of sense for us to first understand the initial 5 groups, then move on to group 6 and group 7. So that's the way we've done things. So you can see the first point of the lecture is titled retroviruses, and there's going to be a big focus on retroviruses today because members of group 6 are retroviruses, and these members are really important potentially to human health. So there are specific viral species that exist within the family Retroviridae, so they are retroviruses that have a huge impact on human health and well-being, and really what we're thinking about here is HIV-1 and HIV-2. There's other retroviruses that impact human health, but HIV-1 and 2 essentially have this massive impact on human health globally. If we think about virology as a scientific discipline, we can actually say that a huge amount of focus is given towards HIV. There's this huge amount of research that's done on this particular subfield of virology, which we define as retrovirology. And for a long time, we can really think of the field of virology as having 2 main principal focuses, these being first, HIV, so retrovirology, and the other being emerging infectious diseases. So these viruses that cause new and emerging diseases, so SARS coronavirus, swine flu, bird flu. These are for a long time been at the forefront of virology. Now obviously there's a huge number of other viruses that cause significant amount of human disease, but it's just these 2 areas have received a huge amount of research attention, so much so that if we look at all the antiviral drugs that have been approved, so that exist, we see that about half of them have actually been developed to target HIV. So about half of them are antiretrovirals, and this kind of represents how dominant HIV is in the field of virology. So this is why it's actually really, really important that we understand group 6, because HIV belongs to group 6. It therefore expresses its genome and replicates its genome in a manner that is different to other single-stranded RNA viruses, and we need to understand these differences because, as we'll see, a lot of the therapies that target HIV infection rely specifically upon the way HIV expresses and replicates its genome. So really important we understand this today. You can see Hepadnaviridae at the bottom, so near the end of the lecture, and it's also really important we understand Hepadnaviridae. We're referring to group 7 viruses here, so the final group on the Baltimore classification system. This includes hepatitis B virus, which of course has a significant impact on human health as well. So it's a really, really important causative agent of human disease. And again, even though it's a DNA virus, it replicates differently to the other DNA viruses, so it expresses and replicates its genome in a different manner, and it's really important we understand that because there's implications for therapeutics, which again we'll talk about in the lecture. And here we have the Baltimore classification system on the left and the phenotypic classification system on the right. Again, these are systems you should be really familiar with by now. If you're not, I'd encourage you to go and listen to the classification system again. And today, as we said, we're talking about class 6 on the Baltimore classification system, which is the group of single-stranded RNA viruses that engage in reverse transcription. Of these groups, we are very interested in HIV, again because HIV has a significant impact on human health by being causative of AIDS, and it's really important we understand the distinction between HIV and AIDS, because we don't want to be going into a level 6 virology exam not really understanding that there's a difference between HIV and AIDS. HIV is a virus, and this virus is the causative agent of AIDS. So HIV infection will potentially, if untreated, progress towards a disease state, and this disease state is defined as AIDS, which is acquired immunodeficiency syndrome. So it's an immunodeficient state which is caused by HIV infection. So really important we understand the distinctions there. This is a diagram of a generic viral life cycle that we've seen before. So where a virus attaches to the surface of the host cell, then enters into the host cell, then replicates its genome and expresses its genome, and the product of genome replication and expression, so the genome copies and also the proteins, these assemble into new virus particles which then leave the cell where they can infect new cells or a new host. Now, this life cycle applies completely to HIV. So HIV, in terms of its life cycle stages, is perfectly normal for a virus. The virus, so HIV, will attach to the host cell surface, then it will enter into the host cells where its genome will be expressed and replicated. Then the products of expression and replication, so proteins and genome copies, will assemble into new virus particles, and these new virus particles will then leave the cell where they can potentially infect other cells in the same host or in a new host. It's important to note that HIV is an enveloped virus, so it has an envelope. That means as the virus particles are leaving the host cell, they essentially steal a small portion of the host cell membrane. Now we call this portion budding because a little bit of the cell membrane kind of buds off, and this is again what becomes the viral envelope. As we know, HIV causes immunodeficiency, which is again the state of AIDS, acquired immunodeficiency. One of the ways in which HIV causes immunodeficiency is by directly causing the lysis of the immune cells that it infects. So this process, this viral replication cycle, is occurring in immune cells. These are the cells that HIV infects, and this process puts the cells under a huge amount of stress. So first, the cell has to redirect a huge amount of energy to producing these virus particles. So the virus co-opts the host cell machinery and essentially forces the cell to begin expressing viral proteins and replicating the viral genomic acid. So this puts a huge energy cost on the host cell. Second, as the new virus particles leave the cell, they are, as we said, stealing small portions of the cell membrane, essentially, and this impacts the integrity of the host cell membrane, which impacts the integrity of the cell overall, because the membrane very much protects the cell from the external environment, right? It stops random particles, random molecules passing in and out of the cell. So by having new virus particles budding off from the cell membrane, this costs the cell as well. This is detrimental to the cell, and often this process ultimately results in lysis of the cell, so essentially cell death. And this is one of the ways in which HIV causes immunodeficiency in a host. It's not the only way, but it's a significant way, and it certainly contributes. The other way, just out of interest while we're on this topic, is HIV causes mass activation of the immune system. So it causes activation of immune cells. So nascent immune cells that are not ready yet, they're not functional yet, are suddenly activated, and HIV then infects these cells, again causing lysis. And even if these cells are not infected, they have a fairly short life expectancy anyway, so they have quite a short half-life. So what this means is that by activating the immune system in this rounded manner, HIV is causing all of these cells to ultimately die because of their short half-life. Ultimately, the immune system runs out of cells to replenish this supply, and this is when you get an overall drop in immune cells. So there's this little lift as HIV activates all the immune cells, then this massive drop because the immune system doesn't have any precursor cells left. It can't keep up with the demand that it is being placed under by HIV. So ultimately there is this drop in active immune cells. And again, HIV directly lysing the immune cells contributes to this. And again, this is what we're saying here. So this is just a few sentences defining this. So HIV infects the immune cells, specifically CD4 plus T cells. So these are immune cells that have the specific receptor that HIV can attach to. And the long-term depletion of these cells leads to immunodeficiency, which is again characteristic of AIDS. Now, you're probably aware, I'm sure, that HIV is a chronic incurable infection. So many viral infections can be cured with therapeutic interventions. Many viral infections can also be cleared naturally by the immune system. So that's what we call when the immune system overpowers a viral infection. We call it clearing infection because it gets rid of the virus from the body essentially. So it gets rid of the infected cells until there's no virus left. HIV is problematic from this perspective because it can't really be cleared therapeutically. We're not able to cure HIV is another way of saying that, and people who have a stable infection, their immune systems aren't capable of clearing it either. So this is problematic because it means HIV is essentially a permanent infection. With HIV being a sexually transmitted infection, there's also a lot of stigma around it. So therefore, this idea of HIV being a permanent infection is really adding to the weight, so adding to the consequences that are associated with HIV infection. And again, physiologically, the consequences of HIV infection are minimal with the current therapies we have. So someone who is diagnosed with HIV can expect to lead a perfectly normal life as long as they are able to take the appropriate antiretrovirals every day. However, there's still a burden associated with it because it's a stigmatized infection, and because, of course, there's a psychological weight to being diagnosed with a chronic incurable infection, particularly one that's stigmatized. This chronicity, so this ability for HIV to infect long term, so permanently essentially, has a biological basis, and this is tied to the ability of the virus to replicate using reverse transcriptase. So again, we're considering that HIV belongs to the 6th group on the Baltimore classification system, and that means it's a single-stranded RNA virus that engages in reverse transcription, so it has reverse transcriptase. Now, one consequence of this is that HIV has the ability to infect permanently. So it has the ability to infect not in a acute manner that is then cleared by the immune system, but infects in a way that the immune system is not able to clear, and again we are not able to clear therapeutically, and this is intrinsically tied again to the way that HIV expresses and replicates its genome, and that's why it's really important we understand this. And the key fact behind this is the observation that HIV can integrate into the host cell genome. So as part of HIV genome expression and replication, it integrates into the host genome. And we touched on this very briefly last week, and what we're doing now is fleshing out our understanding of this, because by integrating into the host cell genome, HIV gains the ability to infect permanently. So it gains this chronicity. So again, this is why we need to understand the process. Okay, so some information there on retroviruses, a little bit of context, a lot of it you may have known already. Hopefully there was a bit of new information in there, and at the very least, it was hopefully a good reminder. Now we want to talk about specifically reverse transcription. So we've said these viruses, these retroviruses, including HIV, have the ability to integrate in the host cell genome, and this gives them the ability to infect with chronicity, so cause long-term infection. And this is tied to their ability to reverse transcribe. So reverse transcription and integration come hand in hand for these retroviruses, and they're both important aspects of genome expression and replication. So this is what we're going to talk about now. First, reverse transcription, then integration. So this is a process we touched on in the last lecture, and what we want to do now is flesh it out a little bit and really understand how HIV engages in the process. So as we discussed, reverse transcription is the opposite of cellular transcription. So in cellular transcription, the host genome is transcribed to produce RNA. In reverse transcription, the HIV genome, which is RNA, is reverse transcribed into DNA. So this is fundamentally what we mean by reverse transcription. And reverse transcription essentially allows the virus to get around this problem of not being able to integrate its genome into the host cell genome. And this is what we mean by reverse transcription and integration being really tightly linked. It would be absolutely impossible for a retrovirus to integrate an RNA genome into the DNA genome of the host. It just doesn't make sense. It just wouldn't work. So for the process of integration to occur and for HIV to establish this latency, this chronicity, it has to first reverse transcribe its genome. It has to first use its RNA genome to produce a DNA copy, and then this DNA copy can potentially be integrated into the host cell genome because it's the same format. So what ultimately happens is the DNA product of reverse transcription is essentially put into the DNA genome of the host, and this is viable. It is able to occur because the format is the same, and again this format is there because of the process of reverse transcription, because the virus has produced DNA from its RNA genome. Really different to anything we've talked about before. Obviously this is a really specific process that is occurring with these retroviruses, and yeah, it's just important that we understand that. And again, we've talked about specific genomic processes in the past that viruses engage with. So obviously we know what transcription is in the host cell, and we've talked about how viruses engage in a version of transcription where their genome is used to produce an RNA transcript which can be translated. So we've said viruses can have different genome forms: RNA, DNA, double-stranded, single-stranded, and whatever the genome format is, they have to produce RNA as a copy of their genome, so this RNA can be translated, and this is really essential to genome expression, the production of protein. And then we also have the process of genome replication that viruses engage with, so using their genome to produce a copy of their genome. Again, this should be an exact copy with the exception of some mutations, and this is why we term it genome replication, because they are copying the genome. And now we're introducing what is essentially a third genomic process. So this process of integration, which occurs after reverse transcription, and this process is occurring in retroviruses. So retroviruses, like all viruses, need to express their genome to produce protein, and they also need to replicate their genome to produce exact copies. However, to allow retroviruses to do both these things, genome expression and genome replication, they need to first engage in this process of reverse transcription and integration. This is just the way the expression and replication pattern of retroviruses occurs. And as we're going to see next, genome expression and genome replication is intrinsically tied in retroviruses to their ability to reverse transcribe their genome and integrate this product of reverse transcription into the host cell genome. So it's best to think of this process, reverse transcription and integration, as being a necessary precursor to genome expression and genome replication that occurs in retroviruses. And again, just to clearly define this process of reverse transcription, we are going from an RNA genome, so the retroviral genome is RNA, they're RNA viruses, and this RNA is being used as a template to produce DNA, and this DNA is what's integrated into the host cell genome. The process here is, again, RNA is a template to produce DNA, so therefore the enzyme that is facilitating this must be an RNA-dependent DNA polymerase, and this is the type of enzyme it is. If we want a specific name for the RNA-dependent DNA polymerase that is utilized by retroviruses, we use the term reverse transcriptase. It's just an easier term to use. We can say reverse transcriptase is an RNA-dependent DNA polymerase, and that makes perfect sense. That's completely true. Again, reverse transcriptase is just quite a useful descriptive name because it's A's means enzyme, and reverse transcriptase indicates that it mediates this process of reverse transcription. So again, the production of DNA from an RNA template. Okay. And at the bottom here, as we've said, this process of reverse transcription followed by integration is a precursor to retroviral genome expression and replication. So in these retroviruses, reverse transcription and integration must occur before they are able to express their genome and replicate their genome. Okay, and again, this is the process, just a diagrammatic view if anyone's struggling with it. In a normal host cell, we have transcription, so RNA produced from DNA, mediated by an RNA polymerase, so a DNA-dependent RNA polymerase. And in retroviral reverse transcription, we have the opposite of the process. So we have DNA being produced from an RNA template, and this is facilitated by reverse transcriptase, which is an RNA-dependent DNA polymerase. The actual process of reverse transcription is really quite complicated. So this is a diagram I made a couple of years ago just because it's such a complex process. The only way to really understand it is to take it apart step by step. And one way to do this is to, create a figure. If you're creating a figure, you need to know exactly where everything goes. I'm not going to expect you to learn this figure. I don't think it's particularly useful for you to learn the specific step-by-step process of reverse transcription. The only real purpose of showing this is to demonstrate just how complicated the process is. It is a really, really complex process, but the key information we need is that the virus is going from RNA to DNA, and we can see that here. We can see initially there's this light blue strand of RNA, then there's a lot of polymerase activity, which is what is represented by the red arrows. So each red arrow is the progression of an RNA-dependent DNA polymerase along the nucleic acid. And there's also a lot of shuffling around of partial transcripts. So we can see partial transcripts being transported from one end to the other, and this just facilitates the movement of the RNA-dependent DNA polymerase along the complementary strand. Okay. And essentially, again, we don't need to worry too much about the individual steps. Definitely don't expect you to memorize these steps. But if we can take on board the key information, which is that initially, as we can see, light blue RNA strand, and ultimately the end point is double-stranded DNA here. It's again complicated, but I wanted to have this here so it seems a bit more real. It doesn't seem like some mystical process by which RNA is turned into DNA. It's not what happens. What happens is there's a strand of RNA, and then DNA is made as a copy of the RNA. So RNA, one strand of complementary DNA is made. This RNA is got rid of, then a complementary DNA strand is made to the first DNA strand, and then as a result there is double-stranded DNA. And we can see that halfway through here, and this is quite key, there is a complex of RNA with DNA. So we can see RNA bound to DNA here, where we have the light blue RNA with the dark blue DNA. Really unusual to see this. We call this an RNA DNA duplex. So a strand of RNA bound to a strand of DNA. Really unnatural, really not normal for this to be seen in a cell. This is only ever really seen in reverse transcription. Now, it follows that an RNA DNA duplex is a very potent inducer of an immune response. Again, reverse transcription only ever occurs in infected cells. This is not a normal process that occurs in healthy cells. And RNA DNA duplexes, so RNA bound to DNA, are intermediates of reverse transcription, as we can see. So in a normal healthy cell, there's never any reverse transcription, so there is never any RNA bound to DNA. Therefore, our immune systems have evolved to specifically react to RNA bound to DNA because it is such a strong indicator of viral infection. And as I'm sure you're aware, our immune systems have evolved extensively to react to anything that is potentially not self, so anything foreign, because anything foreign indicates the presence of an outside microorganism that could potentially be causative of disease. And that's exactly what's going on here. Our immune system has evolved to react to RNA bound to DNA because it's an indicator of reverse transcription. Anyone who's doing a project in the lab with me this year, or who has perhaps heard me talk about my research or read about my research, you may be aware that we can use something called poly-IC in the lab. And what poly-IC does is it essentially induces an immune response in cells in tissue culture that is similar to the immune response we would see if a real virus were used to challenge the cells. The reason poly-IC is capable of doing this is because poly-IC is essentially a mimic of the RNA DNA duplex that is formed in reverse transcription. So poly-IC is an artificial version of RNA bound to DNA. So it mimics the duplex, and when you add it to cells in tissue culture, these cells react as if they were reacting to a real intermediate reverse transcription. The other takeaway is that because this is a uniquely viral process, so this whole process of DNA being produced from RNA only occurs in viral reverse transcription, it means that we can successfully target this process therapeutically, hopefully without having too many side effects. And this is what we actually see. We see drugs that target reverse transcription tend to be fairly low in toxicity. Again, this is because they're specifically inhibiting a viral process. A lot of drugs that are high in toxicity, so they have a lot of side effects, are this way because they target processes that are microbial processes, but maybe they're also host cell processes, or maybe they're microbial processes that are very similar to host cell processes. And this means the drugs not only target the microbial process, but they also potentially target the host process as well. And again, this is problematic. This is how toxicity comes about. And we mentioned earlier that a huge number of antiviral drugs are specifically targeting HIV, so they're antiretrovirals, and a huge proportion of these are specifically targeting the process of reverse transcription. And again, this is because reverse transcription is a very attractive therapeutic target because it is a uniquely viral process. So therefore, in theory and also in practice, so what we actually see is it can be targeted successfully with very low toxicity, again because it's a viral process. Okay, and that's the process of reverse transcription, and as we can see, the product of reverse transcription is a DNA version of the viral genome. So the initial genome is single-stranded RNA, and reverse transcription produces a DNA product that is double-stranded. The process that follows this is integration. So the DNA product of reverse transcription is then integrated into the host cell genome. Again, as we've said, it is only a DNA copy of the viral genome that can be integrated into the host cell genome, because the host cell genome is DNA, and it doesn't make sense to integrate an RNA version of the genome into a DNA genome. It just wouldn't happen. So this is why the genome is first the RNA genome is first used to produce a DNA copy, and then this DNA copy can be integrated into the host cell genome. Okay, and here we have the DNA copy of the viral genome, and now we're going to look at how it's integrated into the host cell genome. And again, this integration has to happen for viral genome expression and viral genome replication to occur. So the first step of the integration process after reverse transcription is processing of the 3' ends of the double-stranded DNA copy of the viral genome. So the double-stranded genome exists, and there are certain changes occurring to the 3' ends of both the strands, because both of the strands have a 3' end and a 5' end, right? And this processing occurs at the 3' end of both, and this process makes each 3' end hyper-reactive. So it makes each 3' end prepared to engage in particular interactions, and essentially the genome is then forced into the host cell genome in such a way that these interactions can occur, and these interactions occur between the exposed 3' ends of the viral genome and the host genome. And this is how it is kind of forced into the host cell genome. And again, the enzyme that is mediating this process is integrase. So as we can see here, integrase is the viral enzyme that mediates this integration. Again, integrase, it's an enzyme, ase, and it is mediating integration. We've mentioned many times that the structure of a virus is the nucleic acid genome surrounded by protein, sometimes surrounded by a lipid envelope. And we also need to consider that with the genome inside the protein shell, there often exists functional proteins. And I always show you this diagram of a HIV particle that I made, and you can see the genome is in the middle and it's surrounded by the protein shell, but there are also these dark red dots next to the genome. These represent functional enzymes. So these represent enzymes that the virus uses during infection. And one of these enzymes is integrase that we just talked about. And another one of these enzymes is reverse transcriptase, the RNA-dependent DNA polymerase that we talked about. So these are two viral enzymes that are obviously really important for viral infection. So for the life cycle of the virus, that's why they're included in the viral capsid. And now we're seeing exactly why they're important, because reverse transcriptase is mediating reverse transcription, and integrase is mediating the insertion of the product of reverse transcription into the host cell genome. Okay, and that's integration. So we're walking through this like a story. First off, we have reverse transcription, then we have integration. And now at this point, we have arrived in a situation where the viral genome in the form of a DNA copy is existing inside the host cell genome. So it's integrated into the host cell genome. And at this point, retroviral genome expression and replication can occur. And that's what we're going to look at now. Some of the concepts we'll see now will be familiar. Some will be slightly different, but we're going to work through them at a steady pace. So hopefully they'll become clear. Okay, so retroviral genome expression and replication. Okay, so just to have the context on the screen, here we have a simplified version of HIV genome reverse transcription, and in a second we'll have integration as well. So here we can see the RNA genome being used to produce DNA, and this process is of course reverse transcription as we've discussed, which is mediated by reverse transcriptase, which is an RNA-dependent DNA polymerase. The next stage is integration. So the DNA product of reverse transcription is integrated into the host cell genome, and this is mediated by integrase. Again, this is what we've established earlier in the lecture. The next stage is what we're now concerned with, which is the genome expression and genome replication stage. Now, as we know, all viruses need to produce RNA to express their genome, so this RNA can be translated into protein. And understandably, the enzyme that is going to be involved in this process for HIV is going to be a DNA-dependent RNA polymerase, because the viral genome is in a DNA format at this point. And again, for transcription and translation to occur, this intermediate of RNA is needed. So transcription produces the intermediate, and then translation uses this RNA intermediate to produce protein. So central to genome expression for HIV is a DNA-dependent RNA polymerase. And as we'll see, the same enzyme is implicated in genome replication. Essentially, for viral genome expression, in these instances, so for HIV, we can think of transcription as being exactly the same as host cell transcription, because the viral genome is in a DNA format, it's integrated into the host cell genome, so the viral genome is expressed just like other components of the host cell genome. And again, the enzyme DNA-dependent RNA polymerase is the same DNA-dependent RNA polymerase that is used by the host cell, because it's the same process. There are some distinctions, however. We talked earlier about the switch between genome expression and genome replication, and we see the same thing for HIV. So there are some distinctions, and these distinctions are between genome expression and genome replication. So for genome expression, HIV tends to utilize subgenomic fragments, so tends to have specific genes expressed. For genome replication, as we talked at length about, HIV requires the production of full-length RNA versions of its genome. And therefore, as we can see, on the one hand, we have genome expression utilizing subgenomic fragments. On the other hand, we have genome replication representing full-length fragments. And each of these processes are obviously mediated by a DNA-dependent RNA polymerase, which happens to be the same. So it's the same enzyme that transcribes the host cell genome. And again, we can see in either case, it's using a DNA template to produce RNA. The difference is whether it is producing full-length genome copies or subgenomic fragments. And what we're going to see now when we look at this process in more depth is we see there's a switch, just like there is for many other viruses. So there needs to be a switch from genome expression, so from subgenomic fragments to the production of full-length genome copies, so a switch from genome expression to genome replication. So what does this switch look like? You'll see there's a new term added onto the slide. So just to introduce this term now, it's provirus. And when we say provirus, we're referring to the viral genome integrated into the host cell genome. So we refer to this as the provirus. It's just a bit easier than researchers or scientists saying the whole sentence of the viral genome integrated into the host cell genome over and over again. So when we say the provirus or the proviral genome, we're referring to the fact that at this stage integration has occurred. And that's what provirus essentially means. Okay, and what we want to consider now is the switch from genome expression to genome replication. So the switch from the production of subgenomic fragments to the production of full-length genome copies. So the process overall should be somewhat familiar in the sense that we're going to see an accumulation of protein that is being produced from genome expression. So genome expression occurs, a particular protein is produced through translation of the transcripts, and then over time, as genome expression continues to occur, the levels of this protein build up, and then a change is induced by the building levels of these proteins or this protein that causes a switch from genome expression to genome replication. So, and by that we mean a change from the production of subgenomic fragments to the production of full-length fragments. Okay, now it's important to note what's happening here is the full length transcript is normally being produced and then spliced up, so cut up into the subgenomic fragments, and this process occurs in the nucleus. Okay, and then these subgenomic fragments are translated, and as part of this translation, Rev is produced. So one of the proteins produced is referred to as Rev. So this is a viral protein produced by translation of subgenomic fragments. As this continues, Rev levels build up, and as the levels build up and increase, Rev protein begins to bind to an element on the full length transcript, so before splicing occurs, before it is cut up, and this element is referred to as the Rev response element, so RRE. Now again, Rev levels are building up within the cell, and once they reach a certain threshold, they begin binding to the RRE on the full length transcript, so before it is chopped up into the subgenomic fragments. And again, by chopped up, I'm referring to the cellular process called splicing. Okay, once Rev starts binding to the Rev response element, it causes export of the full length transcript into the cytoplasm. Now this export, if it happens rapidly enough, happens before splicing can occur. So Rev builds up in the cell, starts binding to the Rev response element, and it induces the transport of the full length transcripts into the cytoplasm, and by doing so it prevents the splicing, so it prevents the subgenomic fragments from being formed. Okay, hopefully that makes sense. Rev builds up, binds to the Rev response element on the full length transcripts. The full length transcripts, once they're bound to Rev, are able to move into the cytoplasm, so they're transported into the cytoplasm, and this means the splicing, so the cutting up into subgenomic fragments, never occurs. And here we have essentially the full switch. This is what's happening because these full-length genome copies that have been exported to the cytoplasm are absolutely viable genome copies. They're full length, they're in the right format, they're positive sense single-stranded RNA, and as we know, HIV is a positive sense single-stranded RNA virus. If you don't believe me, you can look at the phenotypic diagram. It's right there with the other positive sense single-stranded RNA viruses. The only reason it's in a different place on the Baltimore classification system is because the Baltimore classification system is based upon genome expression method, isn't it? So this positive sense single-stranded RNA version of the genome is sufficient to be a genome copy, so a product of genome replication, and is packaged into the new virus particles. Okay, there's also another potential destination for these full-length copies. They can actually be translated into protein as well. So again, there's sufficient genome copies. They can be packaged into new virus particles, but they can also be translated into protein. And what we see is that when these full-length transcripts are translated into protein, different proteins are produced compared to the proteins that are produced from the subgenomic fragments, because there's a different kind of open reading frame. So the ribosome is able to produce amino acids based upon the same genome sequence, not genome sequence, the same nucleic acid sequence, but the sequence is longer and it's able to start at a different point. So if you take one sequence of nucleotides, the ribosome can start on the first 3, so 1 to 3, or it can start 2 to 4, or it can start 3 to 6. And this is how this single strand can produce different proteins. Because if the ribosome starts on the first codon, so nucleotides 1, 2, and 3, and reads all the way along, it's going to read a completely different set of triplets compared to if it were to start 2, 3, 4. Because remember, this is how the ribosome works. It's how it determines what amino acids should be added. It reads codons, so it reads 3 nucleotides at a time. Again, this is cell biology stuff that hopefully we've covered in different modules. If we haven't, I'd encourage you to do some reading up on it. But the gist is that this full-length genome copy, this full-length transcript, can be translated into different proteins from the ones that are produced by the subgenomic fragments. There's a little bit more complexity to introduce because there's actually a middle stage between genome expression that we looked at and genome replication that we looked at. So between the production of subgenomic fragments and the production of full-length genome copies, there is this stage where partially spliced copies can be produced, so partially spliced transcripts. And what is essentially happening is the RNA transcripts are being produced from the provirus, and before these RNA transcripts can be fully spliced into subgenomic fragments, they are exported from the nucleus by Rev. So at this point, Rev levels are sufficient to induce the transport of transcripts out of the nucleus, but not to do so so rapidly that no splicing can occur. So Rev is binding to the Rev response element and inducing the transport of transcripts out of the nucleus. However, it is not doing so rapidly enough to prevent any splicing. So some splicing is occurring and the product is partially spliced transcripts. And these partially spliced transcripts can be translated in the cytoplasm. Now, as we've looked at, these different length transcripts can produce different proteins, even though they have fundamentally the same sequence, and this is true here. So the subgenomic fragments can be translated into some proteins. The partially spliced transcripts can be translated into other proteins, and the full-length genome copies can be translated into other proteins again. This is really, really important because it enables HIV to essentially switch from the production of early genes, and these are the product of translation of subgenomic fragments. So it can switch from the production of these to the production of mid genes, so these proteins that help the middle stage of infection, and these are the products of translation of partially spliced transcripts. And then finally it can switch to the late proteins, so the expression of late genes, and this is the end product of translation of full length genome copies. Okay, so there's a lot of information there. I'd encourage you to go back and read over it, listen over it a couple of times if it's not clear. Also, strongly encourage you to read around the topic. There's a lot of really useful information in various textbooks and a lot of resources online. Okay, and again, I've got that written on the slide just to make it clear. Transcripts can be partially sliced. Medium Rev levels result in the export of partially spliced transcripts from the nucleus. This is before Rev levels are high enough to mediate the export of full transcripts. So it's a kind of middle ground, it's a middle level. And again, as a consequence of having this strategy of switching from genome expression to genome replication, which is essentially a strategy of changing the end product of transcription. So what is the end product of transcription? A subgenomic fragment or partially spliced fragment, or is it a full-length copy of the viral genome? By being able to switch between the two, HIV is essentially able to have these three stages of expression: the expression of early genes, then the expression of mid genes, then the expression of late genes, and it mediates the switch between these stages by the accumulation of Rev. And again, Rev is the viral protein that will bind to the Rev response element on the viral transcripts, and that will mediate the export of transcripts into the cytoplasm. And this is a really effective strategy that really helps HIV infect cells, because the early genes are genes that help HIV to overcome the initial immune response and establish infection, and then the mid to late genes are genes that really help HIV to create new virus particles once infection is established. So it's a really, really interesting system to look at, and again, it's one of the things that makes HIV such a formidable virus. Okay, and again, this is essentially what I'm referring to here. The early genes are establishing the early infection, mid more involved in maintenance of infection, and then the late genes, these involved in ensuring new virus particles can be made, so preparing the next stage of viral infection. And by switching from one to the other, then to the other, HIV is exerting a really interesting level of control, which otherwise it wouldn't have. Remember, when we have cellular genome expression, there are some really interesting control mechanisms there. There are ways we can turn genes off and turn genes on. There's all these molecular mechanisms there. HIV as a virus, it's really simplistic. It doesn't have its own machinery. It hasn't evolved the capability to do things like this. This is one of the ways that HIV specifically has gotten around that and managed to evolve a means to exert some control over its own genome expression. Okay, and that was retroviral genome expression and replication. It is complex. There's a lot of information there. Again, really encourage you to go back and re-listen if you struggle with any concepts. Again, please do consult the literature if you feel the particular way I'm explaining something doesn't really connect with you. Sometimes that can be the case. You'll see literature and textbooks saying basically the same thing I'm saying, but in a different way, and sometimes that can be really, really useful for students hearing the same concept explained in a slightly different way. So you're strongly encouraged to look at the textbooks and other resources if you think it would be useful. Okay, so to finish off, we want to look at the hepadnaviruses, so members of the viral family Hepadnaviridae. These are the prominent member of the class 7 on the Baltimore classification system. So we're now looking at class 7 viruses on the Baltimore system. So we've hopefully established group 6, and what we want to think about now is group 7. Again, this is what we're talking about when we refer to Hepadnaviridae. These double-stranded DNA viruses that engage in reverse transcription. Okay, in terms of their genome structure, they have a gapped genome, so they're double-stranded DNA viruses, but they don't have a linear genome like we tend to think of genomes. They actually have a genome that is linked by one strand. So one strand is circularized and the other is partially circularized. We've actually talked about these before, and we've talked about what happens when these enter into the host cell. And the first thing that happens is this gap is filled in by host cell repair mechanisms. So the host cell has various repair mechanisms to close up any open DNA because it assumes, or I shouldn't say assumes, because it detects any gaps, and these systems repair them, because usually a gap would mean DNA damage, and the cell doesn't want DNA damage. It wants to repair any damage. So essentially what happens is the mechanisms in the cell that would normally repair host cell genomes are repairing the gapped portion of the hepadnavirus genome. And the product of this is just a full circularized double-stranded DNA. As we're aware, RNA is required as part of genome expression. It always is, and this process, the process of production of RNA, is mediated by a DNA-dependent RNA polymerase, because the viral genome is DNA, and it is being used to produce an RNA copy. These RNA copies are circularized, so they originally start out as linear, but they circularize, and then these circularized forms are transported into the cytoplasm. And this is where things get a little bit unusual, because these circularized forms can essentially take one of two routes. They can either be translated into protein, which is fairly normal. We know very well by now that RNA can be translated directly into protein if it's positive sense. As we can see, this RNA is positive sense, so this circularized RNA can just be translated into protein. However, something else can happen as well. The RNA can actually be reverse transcribed back into DNA. So the enzyme that's mediating this is a form of reverse transcriptase. It's an RNA-dependent DNA polymerase. It's taking the RNA template to produce DNA. So it's going from RNA to DNA, and this is what can happen for hepadnaviruses. Okay, and that's it essentially. This circularized DNA will kind of hang around in the cell, and it can be a bit of a problem because these hepatinoviruses, namely HBV, hepatitis B virus, has a degree of chronicity, because even when you treat hepatinovirus infection, so HBV infection, these circularized forms of DNA will essentially hang around for quite a while, which obviously there's difficulties with that. It gives the infection a degree of chronicity that a lot of other infections don't have. It's not the same level of chronicity that HIV has, so it's not kind of an integrated infection. There's no integration of the DNA copies into the genome, so they can potentially be cleared. Okay, and now we've essentially arrived at the end of our journey through genome expression and genome replication for all the different viral groups. We've gone through it at a really steady pace, so hopefully everything is clear. Again, if anything isn't clear, I really encourage you to use the resources that are available to you. Rewatch the videos, look through the slides, use the recommended textbooks, look on PubMed at review articles that are relevant to the topic to give you a new perspective. Some of the concepts are quite tricky, and that means it just takes a bit of time for them to be understandable sometimes, so I'd encourage you to put that time in. There's a little bit of information here that might help you. It's essentially a summary of how the different viruses express their genome. So for the double-stranded DNA viruses, they start off with double-stranded DNA, of course, and then this is transcribed into positive sense RNA, and this positive sense RNA is translated into protein. We have the single-stranded DNA viruses next, and the single-stranded DNA viruses start with a single strand of DNA. This single strand is repaired into double-stranded DNA, and then from this point it's the same as double-stranded DNA viruses. So the double-stranded DNA is transcribed into RNA, and this RNA is translated into protein. For positive sense single-stranded RNA viruses, the original positive sense single-stranded RNA genome is used to produce a complementary negative sense strand, and then this negative sense strand is used to produce a positive strand, and then this positive strand can be translated into protein. For the negative sense single-stranded RNA viruses, the initial negative sense strand is just used to produce a complementary positive strand, and again this complementary positive strand can be translated into protein. For genome replication, double-stranded DNA viruses can go straight from their DNA genome to a DNA copy, so nothing super complex here. For single-stranded DNA viruses, essentially the repair initially occurs, right? So you get double-stranded DNA, and then a single-stranded DNA copy of the double-stranded DNA is produced, and this single-stranded DNA is what is packaged into the new virus particles. For positive sense single-stranded RNA viruses, the initial positive strand is used to produce a complementary negative strand, which is used to produce a complementary positive strand, and this final positive strand is what constitutes a genome copy. For negative sense single-stranded RNA viruses, the initial negative strand is used to produce a positive strand, then this positive strand is used to produce a complementary negative strand, and again this final negative strand is what is packaged into the new virus particles. So all this information is stuff we've gone over at length. I just wanted to include these last two slides because it's a really concise way to kind of jog your memory, if you like. It's a real visual representation of a lot of information, and some students find that useful to literally imagine a concise diagram like this in their head. So I just wanted to put it in. Okay, right, thank you everyone again. It'd be great to go through this with everyone in the tutorial next week, so please do attend and engage with questions, and do turn up with any questions you have of me. So if you think there's anything that it would benefit you for me to explain it again, or any additional information you'd like, just let me know in the tutorial and I'll be happy to answer or provide any extra info. Okay, thank you.